Okay, in this module I will discuss the stem cells as a model system to make you understand the cell differentiation better. Just to recall the definition of differentiation. So, differentiation essentially means differential gene expression, and therefore, when cell A is differentiated to cell B, then one would expect that when a cell A is differentiated to, or a cell type is differentiated to another cell type, and every time I show differentiation I always mark or sketch the nucleus in different shade just to show that two cells have different gene expression. Now, so this some basic theory related to the differentiation was covered in one of the earlier modules. In this particular module I will discuss the stem cell differentiation process, and for this purpose I will also introduce you what is the definition of stem cell, how stem cell is different from other cell types, what are the different types of cell types, stem cell types, to have different types of stem cells, and how they are differentiated to other cell types. So, this is a formal definition. Stem cells are defined functionally as some unspecialized cells that have the capacity to self renew as well as the ability to generate differentiated or more matured cell types. So, it can do two things. One is that I will show you in some of the subsequent slides. So, if you have that one stem cell, then it can produce two stem cells while maintaining the stemness. So, it is simply the proliferation of stem cells without being differentiated, but then each of these daughter cells now can undergo differentiation along certain lineages, and they can finally produce different different cell types, and these cell types. So, let us start with the marking of cell. Now, A to B and A to C. So, here up to this part your stemness is maintained. That means, stem cell is now growing, proliferating from 1 to 2, 2 to 4 like that. Now, when B goes to one specific lineage to produce matured cell type C, or C goes to, sorry, this will be D and this will be E, and when C is differentiated to produce to produce a different cell type E. Now, remember D and E have different functionality. So, although they come from the daughter cells, two different daughter cells of one mother stem cell, but their functionality of D and E is different, and therefore, in principle D and E can form different tissue type also. Now, as we progress through the subsequent slides, we learn more about it, and we learn more about what are the different lineages are possible, what is the different types of stem cells, and so on. Now, stem cells, as you know, that have revolutionized the field of tissue engineering as well as human health care, and many researchers claim that almost most of the life threatening diseases can be treated clinically using stem cells, and therefore, in several countries around the world clinical trials using stem cells are currently underway. And so, in this perspective it is important to develop some understanding on the stem cells and how it can be useful in research related to biomaterials, particularly for bone tissue engineering applications. Okay, now having given this introduction to the stem cells, these stem cells can be defined by three unique criteria. One is self renewal, second one is the potency, and third one is the clonality. Now, each of these term has its own importance. So, self renewal means that it can undergo normal proliferation to produce a number of cells without while maintaining the stemness, and this particular case like this essentially that is possible because stem cells have the replicative capacity. Like any other cell types, stem cells also have a replicative capacity, otherwise you cannot call it a cell. So, essentially it is capable of extensive proliferation while maintaining the stemness, and this is immortal, unlimited and continuous. Now, clonality and potency can be described as follows, like single cell with the capacity to create more stem cells, and in terms of the potency stem cells can be classified into 4 different types. One is totipotent, one is pluripotent, third one is a multipotent, and fourth one is a unipotent. Now, unipotent essentially means that ability to generate only one single cell type. Okay. Uni means one. So, unipotent means ability to generate only one cell type. Multipotent means ability to differentiate into many, but limited cell types. It is not all cell types possible, but limited cell type. Pluripotent means ability to differentiate to almost all cell types. So, that is a pluripotent, and totipotent means ability to different all kind of cell types, so that it can develop or it can mature to a organism. So, these are some of the examples are given. Pluripotent example is that embryonic stem cells, and you know that most of the clinical trials, if it involves embryonic stem cells, there may be lot of ethical issues as well as religious issues concerned. Multipotent is mesenchymal and hepatopoietic stem cells, and totipotent is oocyte or zygote. So, let me discuss things in little bit more details. So, the process of unspecialized cells becoming specialized cells is called differentiation or becoming other cell types that has been mentioned before, and differentiation also means that the acquisition of cell type or the matured cell type with specific morphological, phenotypic and functional features. Now, unspecialized stem cells can give rise to specialized or differentiated cells in response to different signals. Now, these in biology literature or in biomaterials literature, these signals or cues are kind of used very interchangeably, and these internal signals essentially turn on specific genes causing differential gene expression, and external signals means either physical contact with neighboring cells or scaffold or biomaterial substrate which has a different elastic stiffness or substrate stiffness. And also conventional biologists, they use different growth factors or cytokines to trigger the stem cell differentiation, whereas material scientists use different scaffolds or different materials, biomaterial substrates as physical cues to trigger or to drive stem cell differentiation. So, these are two distinct kind of approaches. One is that you can use that biochemical growth factors or cytokines to guide stem cell differentiation. Another approach, which is more engineering or more material science based approach, is to use some elastic scaffolds or biomaterial scaffolds which have a specific elastic stiffness which is much higher than that of the cells or tissues, and to use that as a physical cues to guide the stem cell differentiation. So, this is what has been mentioned in the last slide and it is shown here. So, you have some soluble factors like growth factors and cytokines which can drive the stem cell differentiation, and every time it shows that that this schematic you see that these nucleus are shown in different morphology or different kind of colors. Then you have the matrix molecules or you have that substrates that also can guide stem cell differentiation. So, stemness therefore refers to the common molecular processes underlying the core stem cell properties to self renewal and the generation of differentiated progeny. And it is important to mention also that is the stemness establishes their specialized cell cycles either related to maintaining cell cycle arrest in G1. So, remember that a cell goes to a difference one cell goes to G1, then there is a checkpoint, then S, then goes to another checkpoint, then go to G2 and then finally M. So, if a cell is arrested and G1 cycle itself, then it cannot go to unlimited growth conditions, but however it can now differentiate. Now, those things I will explain to you little later. So, as I said before that there are two different types of stem cells. One is embryonic stem cells like which is like embryos created in during in vitro fertilization or aborted embryos that can be used to obtain that embryonic stem cells. Another one is that adult stem cells, and adult stem cells are available in bone marrow, muscle and brain, and these are like discrete populations of adult stem cells that generate replacements for cells that are lost during normal wear and tear, injury or disease. Another place that where adult stem cells can be obtained is placental cord. So, right hand side that one person's image is provided just to show that different sources for stem cells, where you can get these different types of stem cells that I have mentioned in this slide. There are specific biological methods to isolate mesenchymal stem cells. So, like bone marrow derived stem cells, so you can see that here it is shown like a case of a bone marrow biopsy. Then you take this biopsy needle, then you take it, and then there is these different whole bone marrow cell types like RBC, WBC, platelets, adipocytes, as well as hematopoietic precursor cell, endothelial progenitor cells and mesenchymal stem cells. Now, the complete procedure of isolation of mesenchymal stem cells from using this bone marrow biopsy is not under the purview of this course. However, it is just to shown that it is indeed possible to isolate mesenchymal stem cells in this route. Okay. As I said before that in the context of potency of the stem cells, you have pluripotent stem cells, and one of the that if you go back to that one of the earlier slides where I have mentioned, you have the pluripotent stem cells, and this pluripotent stem cells, one of the example is that embryonic stem cells. So, therefore, embryonic stem cells can be can differentiate to different cell type as mentioned in this particular slide. So, one of the cell type is adipocyte, like which make and store fat compounds, astrocyte that it is more related to the neurons or neural tissue engineering applications, like a type of glia cell that provides structural and metabolic support to neurons. Third one is cardiomyocytes, that is the cardiac tissue cells, and it actually form the heart and called myocytes. And remember, this cardiomyocytes is one of the very difficult cells to grow under normal ambient culture conditions. So, therefore, you need kind of very specialized tissue engineering scaffolds or substrates which can support the differentiation of embryonic stem cells to cardiomyocytes. And also I must mention these cardiomyocyte cells, they can beat like a normal heart beats in the when electric stimulation is given externally to these cells. Fourth one is the chondrocytes, that is cartilage cells, I have mentioned before also, that is chondrocytes. Fifth one is endothelial cells, that is the inner lining of all blood vessels, for example endothelium it makes. Hematopoietic cells that differentiate into red cells and white blood cells. So, in case of person suffering from blood cancer and those kind of cases, it is useful. Keratinocytes, that is cells that form hair and nails. Often you see in that open advertisement for treating hair loss, people use stem cells, and to treat the hair loss essentially they you use stem cells, so that stem cells can differentiate to keratinocytes, and then you can one can have fresh hair and nails. Mast cells, that is associated with connective tissue and blood cells. Neuron, that is embryonic stem cells can differentiate to neurons that can form brain, spinal cords or peripheral nervous systems. Osteoblast, as I said multiple times before, osteo means bone, osteoblast means bone forming cells, and from osteoblast it can get differentiate further to a more matured osteoblast, that is called osteocytes. And remember that osteoblast and osteocytes have different distinct cellular morphology. Pancreatic islet cells, that is produce insulin, that is these can be used to synthesize insulin. So, essentially for diabetic patients this is kind of useful. Smooth muscle cells, this is like muscle that lines the blood vessels and the digestive tract. Okay. Now, just a simple comparison between human adult stem cells. As I said that adult stem cells are have a very limited supply, and it can be isolated from specific areas or specific organs of a human patient, and placental cord is one of the place where adult stem cells can be isolated from. Whereas embryos or human embryonic stem cells are pluripotent cells, and they can differentiate to so many cell types as I discussed in the last slide. So, some of the points under this comparison, that stem cells are hard to access and purify, but however embryonic stem cells once isolated, the cells grow with high degree of proliferations. Adult stem cells, or as I abbreviated earlier, that is ASC, it is mostly multipotent. So, it can differentiate to many cell types, but not all cell types. That is the difference between multipotent and pluripotent. Now, human embryonic stem cells, as I mentioned before, they are pluripotent cells. Then third point is that their chromosomes tend to shorten with aging, but here chromosome length is maintained. Apoptosis may be early, where apoptosis is late. What is apoptosis? Apoptosis is the programmed cell death. As I mentioned while discussing about that embryonic stem cells, that use of embryonic stem cells in any clinical trials or typically in many research projects, that can attract serious ethical issues, whereas if you use human adult stem cells, the ethical issues much less. The last point is that adult stem cells, the functionality is more patient specific, hence less chances of immune rejection. In case of embryonic stem cells, there is indeed large chances or higher chance of immune rejection. So, having given this difference between these adult stem cells and then embryonic stem cells, next I would like to discuss that mesenchymal stem cells, and these mesenchymal stem cells are known to generate tissues. First they have the capability to differentiate to cell types like bone cell type, cartilage cell type, muscle cell type, ligament, tendon or adipose. So, therefore, in a way they have the capability to generate tissues such as bone, cartilage and so on. And in the presence of adequate stimuli, remember this stimulation that is I have mentioned in the context of cell signaling. So, in the context of cell signaling, what I mentioned that you have a one cell types, or let us say this is one cell. So, it gets different signal molecules or different signaling cues like A, B and C, which is required only for survival. But if it wants to differentiate to another cell types now, then what it needs some additional signaling molecules or signaling cues, that is D and E. So, that is what has been again reminded here, that in the presence of adequate stimuli, that means you need all these different stimuli, so that it can differentiate to adipocyte, osteoblast and muscle cells. Remember that the signaling cues D and E, which helps in differentiation, that would be of different types when the MSC will differentiate to adipocytes, whereas different type of signaling cues would be required for osteoblast, and at the same time different type of using the same logic different signaling cues are needed for MSCs to differentiate to muscle cells, that is rich in myotubes. So, essentially muscle cells come, they fuse together, then they form a myotubes. And MSCs are indeed rare, relatively rare in the bone marrow, like 1 in 10 to the 5 mononuclear cells, and detected in bone marrow and other places as is mentioned. So, let me take a little bit more time to explain this slide so that you understand and perhaps you can remember this important slide. This slide is little crowded, so therefore, I need to spend little bit more time. What I am showing here, that is the class of I am trying to describe that this I am trying to describe that differentiation of mesenchymal stem cells. So, you have this is your mother stem cells or parent stem cells. So, here I can draw another cells, and let us consider this nucleus with blue like this mother stem cells. Now, this is the 3 stem cells here and 3 daughter stem cells. So, this is the one stem cell, this is the one daughter stem cells, this is the second, and then from 2 other, then you can produce 2 more. So, then you can produce 2 more. Now, each of these stem cell type now can undergo differentiation along specific lineages. So, first one is that osteogenesis, osteo means bone. So, essentially it will undergo differentiation along these lineages, it will undergo differentiation to bone cells. Now, these lineages in a more simplistic terms I can define like each one of us come from a certain lineage. For example, our father, grandfather, great grandfather. So, it comes from that lineage, and then you carry a specific family name and so on, because you come from that family tree. So, similarly, once you try to understand that how stem cells will differentiate, now each type in a specific lineage you can consider it as a grandfather or great grandfather or father and yourself. So, essentially it comes through that particular lineage. So, when it consider that osteogenesis, the first one it comes transitory osteoblast, like it is not yet matured osteoblast, but is it in a transition stage. Then it forms that osteoblast, that is bone forming cells, and finally that end member of this osteogenesis is osteocytes. Now, if you see morphologically, osteocytes is much more star shaped cells, whereas osteoblast has a specific shape, it is more flattened kind of shape. So, morphologically osteoblast and osteocytes are different, and you should be able to distinguish osteoblast and osteocyte morphology on a fluorescence microscope. Now, along the same line, if you want to understand that chondrogenesis process, that is again to make that cartilage cells. So, essentially the first one is transitory chondrocytes, then comes your chondrocytes, then you go to that hypertrophic chondrocytes, like it is growing now. And so, and these chondrocyte cells essentially make the cartilage, which is also shown here. Along the third one, that is myogenesis, that is that muscle cells it can form this myotube, it can form initially myoblast and then subsequently myotube, and subsequently it form muscles. So, in that process of the myogenesis, the first member you can imagine that is myoblast. So, that is the muscle cell. Now, when these muscle cells grow like 1 to 2 and 2 to 4, then two muscle cells they come together and they have a very characteristic spindle shape, and you see that spindle shape. So, it has a very characteristic spindle shape, and the spindle shape they can fuse, they form a myotube. So, this myotube has a larger length and also has a specific aspect ratio, and that forms a smooth muscle cells. So, along these three lineages I have mentioned, I repeat, like it is osteogenesis, chondrogenesis and myogenesis. Now, let me discuss little bit more on the underlying biological aspect. Now, first stage is I separate it out as a proliferation. Proliferation is where your stemness is maintained, like stem cells are now proliferating to large number of stem cell population, with each of them having the capability to undergo differentiation along a specific lineage. Now, what is commitment? Commitment means once a stem cell is committed to undergo differentiation along osteogenesis pathway, then it will follow that specific pathway. It cannot suddenly go from transitory osteoblast to transitory chondrocyte. So, this kind of thing is not a reversible process. So, I have just marked a cross there. So, once it is committed, then it will ultimately from osteoblast and osteocytes if it is committed along this lineage. So, that is called commitment. Now, lineage progression means like along this particular lineage, now transitory osteoblast will now differentiate to osteoblast and to osteocytes. Now, fourth one is the differentiation, that means it has a differential gene expression, it has a different functionality, it is more matured or more specialized cell type than your initial stem cells. And last one is the maturation. Maturation means now osteoblast also is now matured to a is now matured to a different cell type is osteocytes, whereas myoblast fusion which leads to the formation of the myotube. So, this I think entire thing I have kind of covered in this module, which discusses this differentiation of the stem cells.